What makes you unique? What makes us special? The answer lies within humankind's own knowledge as DNA.
DNA is a molecule that stores all the information needed to build and maintain life, from the genetics of both our parents. DNA makes us who we are by giving us characteristics like our physical traits and personalities.
In partners or small groups of 3 or 4, we did an experiment to experience how scientists research and analyze DNA.
However, before we started the real experiment, our teacher, Mr. Reimer, had us look over some papers containing information that would give us an idea of what we would do in the experiment.
On day 1, we learned how to use the equipment needed for the experiment, including micropipettes. The micropipette is a piece of scientific equipment that is used for measuring, transferring, and ejecting small amounts of liquid.
A micropipette has 6 parts: the plunger, tip ejector, volume adjustment dial, volume indicator, shaft, and an attachment point for the disposable tips. The 4 types of micropipettes are P10, P20, P200, and P1000.
The numbers represent the largest amount of microliters the micropipette can hold. A microliter is a millionth of a liter. After learning about micropipettes, we learned what a centrifuge is and how to operate the equipment.
A centrifuge is a piece of scientific equipment that rotates very fast to separate liquids with different densities. It usually has an electric-powered motor. It is also called a microfuge.
When you put the test tubes inside the centrifuge, they must be equally distributed. We use the micropipettes to draw up the dyes and transport them into our own test tubes, along with the other buffer.
We also created a chart to see that we had evenly distributed the buffer and dyes. With our samples in our test tubes, we used the centrifuge to see if we injected an equal amount of mixtures or dyes into the sample.
On day 2, we started to experiment with watercolor gels. We used an Electrophoresis apparatus, also known as a gel box, 1% agarose gel, micropipettes, micropipette tips, a breaker for used tips, a ruler, 1x TAE buffer, plastic wrap, microtubes, and a marker to label the samples.
After getting our needed supplies, we then began our experiment. We used micropipettes to mix different dyes. After mixing our dyes, we pipetted them into the wells inside of our gel, then used agarose gel electrophoresis.
Agarose gel electrophoresis is a method that separates different molecules based on their charge, size, and shape. 2 dams and 1 comb were in the gel box. We put these in the gel before it formed.
The dams and the comb form ridges. We put the dyes into those ridges. We created a gel made of agarose powder and buffer solution. Buffer is used to run DNA in agarose gel electrophoresis.
Then we poured it into our gel box. After it was formed, we put in different colored dyes. We then continued to plug our gel boxes into the power supply and charge them for 10 minutes, setting it at 100 volts.
Then we measured how positively or negatively they were charged. Molecules having a negative charge migrate toward the positive electrode, while positively charged molecules migrate toward the negative electrode.
On day 3, we did the same thing as day 2, except this time we put DNA into it.
We also used the centrifuge to make sure that we had an accurate measurement of DNA. After we charged the gels, we dyed them with fast blast stain.
We use fast blast stain because it allows the DNA to be more visible so we can see the DNA strands inside of our gel once we place them on the light box.
On day 4, we measured the cuts of DNA. Our measurements were organized in base pairs. Base pairs are a pair of well-matched bases in a double-stranded DNA molecule. Base pairs are formed between specific nucleobases.
Later, an assessment from our teacher was given. We needed to fill out a worksheet about what we learned previously in the experiment. Fields of study related to the science experiment include
Chemistry. Because the samples put into the gel had a reaction when we charged them with the power source, they moved towards the positive and the negative side.
Biology. We use biology to figure out how DNA relates to human life and what DNA is. Mathematics. We use math to find out precisely how much amount of dyes to put on our samples and measurements.
Physics. The centrifuge contains many various generalities of physics. The spinning motion forced the samples to be separated.
Analysis. Analysis was used within the experiment, including recording and observing the samples within the gels, making sure we have the right amount of samples, measurements, and solution within our gel box.